All right, let's get started then. This is Heather Fulton-Bennett's PhD defense. We are entirely on Zoom except for a smattering of people in the audience. Heather has been in the program for a few years and is an algal expert. She is the person we go to when we have any questions about algae. And she's going to talk today about her PhD research. Her official introduction is going to be done by Silke Bachhuber. So let's Silke do her thing.
All right. Hey, everyone. Thank you so much for joining us today. If you don't know me, my name is Silke Bachhuber. Heather and I both started in the same year in the Mengi-Lubchenco lab. Before I met her, I knew we'd be spending a ton of time together, which I thought meant we'd either be super close friends by the end of our time in grad school, or we wouldn't be able to stand the sight of each other. And I'll let you all guess what worked out for us. I will keep this really brief because I promised that I wouldn't cry or make Heather cry before her talk, but I want to briefly highlight her incredible accomplishments. She did her undergrad at the University of British Columbia and her master's thesis at Moss Landing Marine Laboratories, where she studied the ecology and reproduction of Egregia, the feather boa kelp. Her PhD at OSU focuses in on an even funkier group of algae, the corallines. Her research dives, pun intended, into some really interesting and important features of this incredibly diverse order. Her work connects fundamental questions about the biology and ecology of coralline algae with prescient questions about how they will be impacted by current and future anthropogenic climate change, an important component of ecology research today. She is also a talented mentor, collaborator, and colleague. Her work at OSU has been supported by a diverse group of funding sources. She has mentored and supported students through the OSU Undergraduate Honors Thesis Program, the Western Society of Naturalists Mentorship Program, and our Undergraduate Research Scholarship and the Arts URSA Engage program. Her collaborative work has resulted in a number of peer-reviewed publications highlighting both her primary research as well as synthesizing key policy statements on marine protected areas, international ocean protection commitments, and more. As a member of the Ocean Science Innovation Team, she worked with Jane Lubchenco to communicate the importance of ocean science to a wide array of audiences. I have learned so much from Heather about how to be a better scientist and collaborator during all the time we spent together back to back in an office, in the car, and dodging waves on rocky shores all around the world. As those of you who have worked with her know, her advice on topics big and small is well thought out, considerate, and always a little bit funny. Finally, Heather is also a talented photographer and naturalist, which made making this slide challenging for me because she's always the one behind the camera. Her passion for experiencing the natural world is clear in the careful attention she pays to even the smallest blade of algae in the field. This is reflected in the work that she does and will share with you today, as well as her leadership positions on both iNaturalist and eBird. She is now joining us from NOAA's most remote field station at Little Port Walter on Baranof Island in Alaska, where she has leveraged her experience, strengths, and interests to have the opportunity to work in one of the coolest places I've ever seen. Please join me in a quick round of Zoom applause before she starts her talk today, titled The Effects of Ocean Acidification on Coralline Algae and Their Associated Communities. Go Heather.
Thank you for the introduction, Silke.
Things are ready.
Oh, sorry. Did I hear that, Bruce, we're all good to go?
Yes, we're ready to go.
Okay,
good. Go.
Yeah, thank you guys all for joining me here today. As Silke said, I am in remote Alaska, so there may be some few interruptions, but I will do my best to pick up where you guys lose me. And first I want to acknowledge that this work was done on the traditional homelands of the Ngaitahu, Ngatirarua, and Ngāti-Toa Rangatira of the South Island of Aotearoa, or New Zealand, as well as the lands of the Confederated tribes, the Siletz, Umatilla, and Grand Ronde of the Oregon coast. And I am giving this talk for the past, present, and future homelands of the Tlingit people of Southeast Alaska. So today's talk, I'm going to give a brief introduction to oceanographic variability in coastal seas and the importance of that variability, as well as coral and algal ecology in a changing ocean. And then going through my data chapters and experiments, looking at coral and recovery from physical disturbance across an oceanographic gradient, coral and diversity across that same gradient, as well as the impacts of acidification on invertebrate settlement queues to coral and algae, and then some overall conclusions. So a lot of times we hear in the news about global change with increasing temperatures, rising seas, ocean acidification, but these changes aren't uniform across our oceans. And we're becoming increasingly aware of the impacts of regional variability and these extremes. For example, marine heat waves and areas of hypoxia that are increasing but are not changing the global baseline in their very short term. This variability is highest at the eastern edge of ocean basins, and this is a naturally variable system due to coastal upwelling. Coastal upwelling is a combination of currents and winds that pull deep, cold, nutrient water to the surface, producing extremely productive environments. But these variable systems are also important natural laboratories for studying how changes in ocean conditions affect marine communities. And the variability in these systems is quite wide, considering our global ocean pH is currently 8.1. But you can see in this plot here how pH is changing and is forecast to change in the future. This is the west coast of the United States, California, and Oregon. And you can see the forecast pH is lowest along the coast and is going to be continually is forecast to decrease in the future. While this overall average is going to change, we are also going to see seasonal shifts in this pattern. So many organisms rely on this upwelling of cold, nutrient-rich waters to feed seasonal blooms, and the phenology of that is also likely to change. So zooming in a little bit more to Oregon, pH is already below 7.8 as much as 20% of the time. Again, global pH is 8.1, and pH is log scale, so that's quite a big difference. And the duration of these low pH events is likely to increase in the future alongside decreased oxygen availability and warming surface waters. We're already seeing negative effects from increasing hypoxia. These are some dead zones from 2006 and 2007 that resulted in really big fish kills of crab and fish along the Oregon coast. And we're also seeing negative effects of decreasing pH in shellfish farms and nearshore environments. So the effects of this low pH in the ocean on organisms is primarily seen, but not entirely, on calcified organisms. And this figure here is kind of the classic of a pteropod shell in low pH, and calcification becomes much more energetically expensive, and at very low pH levels, these shells actually start to dissolve more than the organism can create the shell. So this can have strong negative effects on the organisms. I'm going to focus today on crustose coral and algae. These are red algae in the group Corallinophysidae. I'm going to call them CCA for short because it's a little bit of a mouthful. These are calcified algae, so they have a shell, kind of like you'd think of with a seashell, but it's part of these photosynthesizing red algae. Intertidal down to 270 years.
We lose you right at that crustose coralline algae slide. You here as well?
Still pause?
Not anymore. We lost you at that crustose coralline algae right at the beginning.
Okay. Yeah. So I will refer to these as CCA for short. It's a bit of a mouthful. These algae are found globally, and yes, they are all pink and purple. They're found from coral reefs to Arctic kelp forests and the intertidal down to quite a deep level. They're important ecosystem members as foundation species, literally, at the bottom of a lot of things. But they're also important in cementing together cobble and coral reefs to stabilize reefs, and they're important habitat, food, and settlement substrate for many juvenile invertebrates. And corallines come in quite a range of morphologies. I'm going to be focusing on these crustose forms. They're the most widespread, but there are also articulated forms that have joints, and these rotolith or marl forms. And even these crusts that are quite cryptic come in a really wide range of morphologies. And so take a look closer next time you're in the intertidal and see if you can, guess if there's different species and some of the diversity here. Because corallines are calcified, they have high magnesium calcite. They're quite susceptible to ocean acidification. And as I mentioned earlier, as pH decreases, calcification will become more energetically expensive. In corallines, this results in decreased thickness of these crusts, decreased skeletal strength, decreased growth, decreased recruitment of new settlements. So when the spores settle, it's a lot harder for them to settle and then grow into new corallines, as well as increased mortality. Studies have suggested that coralline abundance globally is predicted to decline by up to 90% by 2100. Given how widespread these are, that's going to have some pretty big impacts. So I'm going to start with my first chapter here, and I'm going to want to look at the differential responses of coral and algal communities to disturbance across an oceanographic gradient. I want to start with thanking my funding sources, the Mamie Markham Research Board from Hatfield, as well as some discretionary funds from Bruce Mengi. I had a bunch of help in the field, as well as help from Beth Davis on my photo analysis methods. So I'm looking at intertidal coralline algae, and the intertidal is a highly dynamic and disturbed environment. We have waves crashing, rocks rolling around, but it's also a highly productive environment. As I mentioned earlier, these coastal areas of upwelling also undergo a lot of oceanographic shifts in pH, temperature, oxygen, nutrients. And in this area, coralline thrive, which is quite impressive for such, so much grows in this area. And so it's amazing what they can withstand. But there's also a lot of disturbance in the intertidal, with rocks bashing around, you get a lot of damage to things growing on the rocks. And so I was interested in how this really highly disturbed environment, how corallines were able to recover from that, given these long-term stresses of changing ocean conditions and these short-term stresses of rocks and physical disturbance. So coralline growth and recovery has been studied, but the focus has mostly been on articulated corallines and then crustose corallines on coral reefs in warm water conditions. Given the wide range of corallines, it's really important to understand how these foundation species can recover from disturbances, even in temperate and cold waters. I should also note that with changing climate, we're also expected to see changing or increased extreme weather and storm events, as we've probably all been experiencing this winter. So these physical disturbances are likely to increase as well as the changing ocean conditions. So to look at this change, or to look at the effects of ocean conditions on coralline growth and recovery, I use a natural oceanographic gradient using the coasts of Oregon and New Zealand. They're similar latitudes and some similar temperatures. So Oregon is an intermittent upwelling zone with upwelling more persistent and more consistent toward the south. Whereas New Zealand, I looked at the South Island, I used the South Island, and the West Coast is a persistent downwelling zone, while the the East Coast is a persistent downwelling zone, while the West Coast has more periodic upwelling is more similar to Oregon in that sense. So I had 11 different sites across Oregon and New Zealand. You can see them here. We've got similar latitude, but and a decent spread across both locations. So for consistency through this talk, when I'm talking about comparing Oregon and New Zealand will in New Zealand, the site and left. So they'll be the northernmost sites in New Zealand. So talking a little, the west sites I mentioned are periodic upwelling. They are actually quite warm and they have a stable high pH and a wide continental shelf, whereas the East Coast are more downwind dominated. The pH is lower than the West Coast, and temperature is more variable and decreases further south. I'd hope to have a southern West Coast site for a little bit more of a colder and a different oceanographic regime, but the weather was too extreme for me to reach it. 3 out of the 4 times I went to south, moving southwards, upwelling is more persistent, pH variability decreases, and temperature also decreases. So my questions were, how does the speed of recovery differ between all these 11 sites, and how does that speed of recovery correlate with oceanographic conditions? I was also interested in how coral and algae are able to recover from disturbances. Is it through encroachment of existing colonies from the edges of a plot in, or is it through new settlement, new corallines settling? So I used these experimental plots to look at that. My expectations were that recovery of coralline would be highest in areas with lower upwelling frequency because of that stress of low pH brought by upwelling. And I was expecting that because of slow growth and the stress of upwelling on new coralline recruits, that coralline recovery would occur mainly through encroachment of existing adult coralline crusts. So my methods, I used a circular quadrat to allow me to look at encroachment. And I installed this in coralline dominated areas of the intertidal across these 11 sites. I photographed these quadrats for 12 months in New Zealand and 18 months in Oregon, and photographed them and then analyzed these plots for percent cover, encroachment, and the community using PhotoQuad. So for a quick example, this is a clearing in Oregon, and this is how it progressed across the course of the 18 months. So you can see it's quite variable. You've got sand incursion, you've got fleshy red algae, you've got massive changes, but goes from almost no corallines to quite a few. So my results. In New Zealand, I found that this is, so here the gray plot is the cleared plots. So these are the areas that I removed all the corallines from at the beginning of the experiment, and the pink is the control plots. Again, the sites are with the sites in the north at the top, and the sites to the south at the bottom. And this in New Zealand, that means the West Coast sites are at the top. And this is showing percent cover after 12 months. So clearings, the gray bars, started at 0% cover at the beginning. So after 12 months, 3 of these sites had actually reached the same percent cover as the control plots. So I would consider those recovered after 12 months. However, in Oregon, which is after 18 months, only 1 site had reached the same percent cover as the control plots, which I was really surprised to see. But also, this is quite, considering we usually think of crustless corallines being quite slow growing, I was also impressed that we had sites that recovered at all, because 12 months is not a long time in terms of these calcified organisms. So for context, I did see recovery being highest in New Zealand and mostly at those West Coast sites. And those are also the warmest sites in New Zealand. In Oregon, the only site that recovered was an intermediate site, so there was not a strong latitudinal change there. So I mentioned growth rates. So I looked at average growth per year. This is in square centimeters. And again, the growth was highest in northern New Zealand on those West Coast sites, and lower and more variable on the East Coast and the southern sites. And in Oregon, it was a lot more variable, but growth was actually highest in the central and the northern, but there was quite a lot of variability. And these 2 northernmost sites that are the extremes are actually less than a kilometer from each other. So it seems to be much more variable. And again, for the temperature decreases to the south, so you can see the gradient in New Zealand of growth, but in Oregon, it's quite a bit more variable, but those southern sites are quite low growth rates. So the last thing I looked at was the mode of recovery, and I used the number of CCA patches in a plot as a proxy for the mode of recovery. So if I have a lot of really small patches, I'm assuming that's due to new settlement of coralline spores that are growing into new coralline crusts, whereas large patches would be a result of encroachment from the edges of the plot, and there would be fewer of those. So in New Zealand, I found that it was pretty scattered. There were quite a few sites with a high number of patches. But 2 of the sites, WB and BT, had quite low numbers of patches. So there wasn't a strong trend here. And again, for a note, the two West Coast sites at the top are not very far from each other. They're about 30 kilometers from each apart. And you might note here that the clearings are space for new recruitment, and patches appears to be lower in the established communities. It's generally higher up here.
Can you start again the disturbance results? That's where we lost you.
Let me know when you can hear me again.
So I can hear you again. We can hear you again.
I'm back. Am
Can you hear us?
I back?
Yes, you're back.
Yes. So for summary, sites in New Zealand recovered more than the sites in Oregon, despite Oregon actually having more time to recover in terms of my comparisons. I saw the fastest growth on New Zealand's west coast, the slowest growth at cooler southern sites, but the mode of recovery was variable across the sites and did not have a strong trend. So going back to my hypotheses, I'd found that the recovery of coralline was highest at the warmest sites and those with wider shelf width, but not necessarily the sites with the lowest upwelling stress. So New Zealand's west coast had the fastest growth and the highest recovery, but that's an upwell. Those sites experience upwelling, but they also have more stable warm temperatures. I found that coralline recovery occurred through both encroachment and settlement and didn't seem to be determined by large-scale oceanographic processes. Given these results, I suspect nutrient availability from upwelling is likely to offset the stress of pH in these conditions, but it's not a simple relationship here. So some of the consequences of these results are that some of these sites may actually take up to, looking at the growth rates, may take up to 10 years to recover. So that's quite slow, thinking about physical disturbance in the intertidal and how often that happens. These organisms are also long-lived and relatively slow-growing if you compare it to, say, fleshy algae or kelp. And there may be a lag in their response to oceanographic shifts. So we may not see negative impacts on them for years as global change, as broad-scale global conditions change. We also don't really understand how some events like marine heat waves are going to leave lasting impacts on these communities. I should also mention that herbivory is another consideration. As populations of herbivores and grazers change, that will affect how these coralline communities are able to recover. So I'm going to switch to my next chapter, and you may have noticed that I have been talking about corallines as a group. And so I decided to look at crustless coralline algal diversity across the same oceanographic gradient following physical disturbance. And again, I want to thank the Markham Award for funding this, as well as the OSU Zoology Research Fund and discretionary funds from Bruce Mengi. Again, I had a ton of help in New Zealand and Oregon, and then a lot of help from folks at Hatfield, especially in the Banks lab, as well as help with all the data. And I'm not a geneticist, so I really appreciate all the help I had on this chapter. So I mentioned that corallines can look quite different, but these are extremes, and coralline identification is really crustless coralline identification is very, very difficult in the field. The identification relies on reproductive material. You can see these little dots on the crust here. But that reproductive material is not always present and is in some species almost never present. And also with microscopic. So just as an example, here's a key from New Zealand on how to ID crustose corallines, and it relies on reproductive structures. But you can see, note here that if you don't have reproductive structures and the plant is not one of 2 species, further ID is just not possible. So that means using genetic marker or genetic techniques. So I took samples of these crusts inside and outside that same clearing experiment I just described, before and after that clearing, and before and after that 12 to 15 months across all those 11 sites. And the reason this coralline diversity is important is that there are many species-specific responses that we are not aware of, and these are also cryptic species that have species-specific interactions with other organisms, either algae or invertebrates. And so understanding their diversity and the community composition can be quite important to understanding intertidal dynamics. So I was interested in what species are found here. We have species lists for Oregon and New Zealand, but given the difficulty for identification, I suspected that that would be an understatement of diversity. I was also interested in how this physical disturbance that I initiated would change species diversity, and whether that would differ along that oceanographic gradient of Oregon and New Zealand coasts. So for this experiment, or for this study, I took chip samples from these same experimental plots. And just to give you an example of the number of samples I was working with, and have the numbers along each step at the bottom here. So I started with 1,500 samples, and then used a DNeasy plant kit to extract and amplify using two different sets of primers to get as much out as I could. I ended up with about 1,100 samples that successfully amplified. And then I did a Sanger sequencing protocol, and then had to go through all the data, clean up the sequences, and I ended up with 924 sequences. And I then sent that off to NCBI's GenBank nucleotide blast system for species ID. So this is what you get from that search, and I ended up with 550 matches that sequences that matched over 95% to a species listed in GenBank. Which is pretty amazing given that there may be undescribed species that are not in GenBank, but I don't have the capability to look at that in this study. So, these are the species. I found 36 species across 11 sites. Way higher diversity than I expected. A lot of these species were undescribed. You can see them here noted, but that was way higher diversity than I was expecting. So when that's broken down to site level, New Zealand on the left, Oregon on the right, you can see the average is around 9 to 10 species per site. And this is in crustose corallines. I wasn't targeting anything else. And the amazing thing is I had 18 species at one site. That was way, way higher than I was expecting. In Oregon, you can see a slight gradient. Those southern sites are on the right, and so there is lower species richness there, whereas New Zealand is quite variable. So I was interested in how diversity changed following disturbance. So I looked at the average number of species per plot in New Zealand and Oregon before and after this experiment. And in New Zealand, so this is the average number of species per plot before and after, with clearings in black, controls in pink. And there was no difference between clearings and controls, which I was a bit surprised about. And diversity did not change significantly. There was a slight upward trend following the clearing experiment. However, in Oregon, there was a dramatic increase in diversity across the course of the experiment, but that was seen in both clearings and controls, but clearings had higher species diversity than the controls. So quite a big difference between New Zealand and Oregon, which I was also surprised about. So the increase in diversity, I believe, may be due to the clearing opening up more plots. That's kind of what I talked about with disturbance, is that when the intertidal is such a rich environment that there isn't always bare space. And I think that Oregon may be more space limited. So the disturbances or the clearings I created were more important in opening up space for new species. I also think that microhabitat may be important, thinking about the range of diversity of species richness in New Zealand was quite large. And I want to note that that highest site that had 18 species, that was a site on the north end of the East Coast, and that is a site that for fish and invertebrates is known to have an overlap of southern and northern species. But there's also a lot of other factors influencing the diversity I found. And I want to talk briefly about the context for this, in that there was a recent study of genetic corallines, and they did a very thorough survey, found almost 1,000 or more than 1,000 species in New Zealand using genetic techniques very similar to what I did here, and most of them were undescribed. And thankfully this paper came out recently, because otherwise I would have had way more sequences that did not have 95% matches. And for context, my sites fit in pretty well. They fill in some of that central area that is a bit undersampled. But they also noted that they found up to 17 species at some of their sites, and that's what I found as well. So I don't think that my high diversity is a fluke. There's also a lot more species to be found. So using that study in New Zealand, species accumulation curves have found that really we still really don't know most of the species there. For context, Oregon is probably better surveyed, but the genetic work, there hasn't been a recent thorough survey the same way there has been in New Zealand that I'm aware of. So in conclusion, coralline's way higher diversity than we think of, especially for something that looks like a piece of bubble gum on the rock. And my sites, I found that diversity was generally higher at the northernmost sites, and it increased after disturbance in Oregon, but not significantly so in New Zealand. One surprise was that there were 10 shared species between Oregon and New Zealand, which is interesting given that they release spores that are non-motile and have very low dispersal distance. So how are those very widely distributed? So I'm in for a second. Now that we've talked about coralline communities across this wide geographic range, and look at how these coralline communities interact with other species. So I'm going to focus on abalone as one of the species, and look at how does ocean acidification affect larval abalone settlement success to crustose coralline algae. And yes, these are big abalone. Again, I want to thank my funding sources, especially the ODI graduate student award. Big Northwest Shell Abalone Company, who provided me with the abalone for this study a long time. So corallines are widely found and globally important, and they have a lot of interactions with invertebrates. The adults will graze the coralline algae, and they'll remove fouling algae and biofilms. But the coralline algae also provides a settlement queue for larval invertebrates, and the presence of corallines can induce the metamorphosis of these pelagic larvae, so they settle from the plankton onto the benthos. So there's positive and negative impact or interactions between these species. So I'm going to zoom in on these settlement queues because they can be quite critical in the success of these invertebrates in, their life stage or their life cycle. There's a lot we don't know about these settlement queues. They are thought to be GABA-based or gamma aminobutyric acid based, which is found in the cells that corallines slough off the top of the crusts that can induce settlement. These cues can also be extremely species specific to both the coralline algae as well as the receiving invertebrate. Some invertebrates have been found to have preferences for specific species. And some of these signals, these chemical signals have been detected from biofilms associated with corallines, but that's very difficult to isolate from the coralline signal itself. So these settlement cues are used by corals, urchins, chitons, abalone, even some sea stars to find good settlement substrate. In corals, there has been some studies on treating coralline algae, the settlement substrate, with low pH conditions, and that resulted in the 86% reduction of coral settlement to the coralline algae. And that is not treating the coral larvae with any kind of low pH. That's just the changes in that signal from conditioning corallines in a low pH condition. And that's dramatic, and that's going to be a major problem on reefs. It's not seen in every species, but it's of major concern right now in terms of the recovery of coral reefs from bleaching and all sorts of the other challenges faced there. Not much work has been done in temperate systems. So that's one reason I was so interested in this, is that we have a lot of invertebrates that rely on coral and algae, and very little has been done to study the interactions. So I used red abalone, Halios rufescens. It's the largest abalone in the world. It's commonly farmed, and they're fairly long-lived, so mature 3 to 7, but they can live for up to 50 years, and they're found along the US West Coast from Oregon down to Baja California, Mexico. I'm going to be focusing on this stage from the velo larvae of settlement to that juvenile stage. And I was interested in conditioning the crustose coral and algae to low pH conditions, and I was expecting that, similar to the corals, if the corallines had been raised in low pH conditions, the abalone settlement success would decrease because that settlement hue would be diluted or changed due to that low pH. I was also interested in how variable pH could have a negative impact on this settlement hue. Some studies have found that variable pH is actually more stressful than constant pH because organisms don't have the time to adjust their internal chemistry to that stressor before it then flips back to the original. So I used a mesocosm experiment that used 3 different pH treatments. I had an ambient, which in Oregon was about 7.9, an extreme low pH condition of 7.3, and then a variable condition where the corallines were alternated between 7.3 and 7.9 every 3 days to kind of simulate that upwelling switch back and forth that we get with seasonal winds off the Oregon coast. And I think this mesocosm is also a little bit different than many studies in that it takes advantage of natural variability. So I was changing the average pH, but I was not changing the scale of variability around that average. So I wasn't keeping it all at a constant pH for the entire experiment. Which is far more natural, but there aren't very many studies that look at this because it's a little bit, most mesocosm studies aren't set up to look at variability. So my experimental design to test the effects of conditioning, I did a range of methods for having the seawater or having the corallines in this experiment. This was an 80-day conditioning, so I kept the corallines in vials for 80 days in these pH treatments. And I had vials with no corallines, vials with corallines, vials with corallines where they were embedded in the Z-spar, and I was hoping to look at growth. Didn't do that, more on that later. And then I
Can you hear us?
All right.
There she is.
Thanks for your patience. Let me bring up my slide again. Where did you lose me?
The Z-spar design, experimental design thing.
Okay, so go back one. Okay. 1 second. Okay. So the reason I did such an expanded Z-spar here is that I did a preliminary experiment and found an effect of it. So I had to expand my controls and disentangle the effect of the Z-spar and the pH treatments. So this is a photo of the mesocosm, and I did not expose the abalone larvae to the pH. This was just for conditioning the crustose coral and algae for that 80 days. And after that 80 days, I switched back to ambient seawater for everything, and then added the abalone to the vials. And again, this was in isolation. Excuse me. This was in isolation because of abalone quarantine procedures in Oregon. So the mesocosm physical conditions. This is just for note. I was mostly want to highlight this central slide of the pH measurements, and that I was able to maintain the pH treatment separately, but you can see that I still had quite a lot of natural variability in that pH level, and you can see the variability obviously in all the other physical conditions as well. So over the course of the 80-day experiment, this is looking at days in settlement across the 17 days after the 80-day experiment. This is the 17 days post-conditioning where I added the abalone, and this is days in settlement across the x-axis, and the percent larvae that had settled on the y-axis. And you can see the pH treatments in different colors here, with high PCO2 in red, low PCO2 in blue, and alternating PCO2 in yellow. And I should have been consistent, but low PCO2 is the ambient condition, and high PCO2 is low pH or extreme ocean acidification. And this is with CCA only. There's no difference in the pH conditions here. But for the CCA that were embedded in Z-spar, there is a difference in the pH treatments, but you can also see that the percent larvae settled really, really decreased when the CCA was embedded in Z-spar. So this is where I had to go disentangle this. So looking at final settlement success, so this is at the end of the experiment when all the larvae should have settled. When just CCA were present, there was no difference across pH treatments, which is great. Really happy to see that for the abalone's sake. But when you have Z-spar present, there's a really marked decrease in settlement success compared to the CCA that were not embedded in Z-spar. And then to disentangle that even more, when you have Z-spar with no CCA, that's even worse. You get even less settlement. But what happens if you have a vial without CCA and without Z-spar? I actually found settlement was still really high. So there wasn't a big difference between the presence of CCA or not under any pH conditions or for settlement, which is kind of wild. And so one last control I added was these vials that had either Z-spar or no Z-spar, and then had not been conditioned under those pH conditions for 80 days. They just, they're fresh, dry vials that I added ambient seawater to, added abalone to, and then looked at. And those are in gray here. So on the far right and then in the middle, the CN condition is an empty vial with no Z-spar, and you can see there's some settlement, but it's a lot lower than those that have been sitting in seawater for 80 days. And then if you have a vial with just Z-spar, absolutely nothing settles. So kind of complicated in that settlement success was highest in vials that had been acclimated to, or like had been in seawater for 80 days, but it didn't really matter if CCA was present or not. So it appears that having those vials in seawater is actually more important in this case than the presence of CCA at all. And what I think likely happened here is that it was actually the biofilm that was more important. The biofilm that developed in the vials after that, during that 80 days. Unfortunately, biofilm components are really difficult to identify and isolate, and I have to admit that microbial ecology is outside my scope. But settlement cues to biofilms that grow in red algae are documented in some species. Not so much in abalone, but it's possible. But we don't really know what the effects of OA on biofilm settlement cues will be, and that's something to consider in the future when looking at effects on these settlement cues and potential changes to this species interactions. So the results were a lot more complicated than I was expecting. There's also the potential for species-specific interactions, given that I did not talk about what species I'm using, and the abalone may be more specific. However, similar studies on red abalone in California, as well as other abalone in New Zealand, have not found effects of OA conditioning of CCA on settlement success. And some of that, I'm wondering if that's due to the fact that these CCA I'm using for this experiment are really used to variable conditions. They are from the Oregon coast. But the decline in corallines or coral settlement success to corallines one factor may be that tropical systems are not as variable as these upwelling areas. The CCA are not used to the variable conditions that then experiments are used. So I mentioned species-specific interactions. I sequenced some of these samples I used for this mesocosm using the same methods of chapter 3, and I found I used these 4 species. Interestingly, only the first 2, Basiella mayi and Exarticulata, are crustose species. The other 2 are actually articulate species, which is, again, surprise, surprise. Also, none of these if you know anything about corallines, a lot of the times people think about Lithophyllum, Lithothamnion, these classic crust names, and that's not what I was finding, which was a fun surprise, but also brings up the question of species-specific interactions, and that maybe the abalone don't love these species, and so it didn't really matter what else I did to them. So on the broader scale, the good news is low pH does not dramatically lower success rate of abalone to corallines, and the fluctuating pH didn't look any worse than the extreme pH. Bad news is that both coralline algae and abalone on their own will still be negatively affected by OA, and that we need to be really aware of how we're using Z-spar, especially in these contained laboratory experiments. The Z-spar had been cured for over 80 days, and there was still a marked negative effect, and it's very commonly used in marine ecology, so we need to be aware of that. We use it commonly in the field for a lot of things, and that's something that it's not like we should necessarily stop using it, but just to be aware and make sure that it's controlled for as best as possible in future experiments. And just as a note, coralline cover will also still be decreasing in the future, so this settlement substrate will decline no matter what happens to the settlement queue. So with this nice water break, if you guys still have me here, I want to zoom out and talk about, again, kind of summarize my overall results. So going back to that first disturbance experiment, I found that sites with upwelling and stable temperatures recovered from my experimental disturbances fastest. And recovery occurred through both encroachment and new settlement, and there was not a strong pattern in which sites had which recovery mode. Then looking at the genetics of coralline communities across that gradient, I found really high diversity, which increased in following disturbance in Oregon, but not in New Zealand. And then the mesocosm, I found no effect of OA conditioning of corallines on the larval abalone settlement success. However, Z-spar epoxy did have a negative effect on the abalone larvae, so we should be careful of that use. So a little bit broader, the negative effects of OA, especially in upwelling areas, may be offset by nutrient availability, which is really great to know, but like they're still vulnerable on coral reefs. That's not Can
you hear as well?
these may interact and what can offset things like global prevalence of coral and algae in all these areas. It's also foundation species like this, because we may not see immediately, and there may be a lag in species like this in results, but I also still have a lot of questions left. And I'd love to look at the successional pathways of these disturbed areas and looking at how these, how that successional community in this areas changes over time, especially with seasonal differences. Given the length of my experiment, there may have been blooms of algae that I didn't capture in the results I showed you today. It'd also, I'd love to look at more into the nutrient availability and how that can offset pH stress, and what's the most important factor there. Also, I mentioned that there were 10 species in common between Oregon and New Zealand. yet we don't really have a great understanding of the dispersal, especially over long distances. And that would be really interesting to look at using molecular techniques and species diversity and markers. And then going back to, of course, my love of kelp. There's a lot more to be done on coralline and kelp interactions in the subtitle, especially when you think about urchin barrens and the importance of that understory community as foundations. And with that, thank you for your patience through the interruptions. I want to thank all the folks who made this all possible. I had a really great field team and yeah, a ton of support over the years. Firstly, to my committee, Bruce Mengi, you brought me into the lab all those years ago and straight into the field, which is exactly what I wanted. And I've never had an advisor to work in the field with before, and that was pretty special. Learned a ton on the rocks that way, especially when I needed to move for waves and when I didn't. And then the rest of my committee, Felix, thank you so much for all the help with the genetics. It's been awesome chatting with you over the last few years. And thank you for taking your Friday to talk from your Friday in New Zealand to talk to me today, or to help today. Sarah, my algae help. Yeah, thank you so much for all the feedback, especially in my experimental design in the early stages. Francis, you've always made me think about the big picture, whether I was comfortable with it or not. And Julia for always being just so reassuring and helpful when I was talking story and trying to figure out what my message would be. And I want to note that Chris Langdon was on my committee for the first part of my PhD before I added a genetics chapter, and I want to thank his help for, he's great making me think about larval dynamics. And then I want to thank the Ocean Science Innovation Team. This is a group that I worked with Jane Lubchenko and a lot of other wonderful ladies on ocean policy issues and learning about policy at the absolute highest level. I learned a ton, and it was a great group to work with during my time at OSU. I also want to thank the integrated biology staff. They were super helpful, especially the T's, the Limengi lab staff who dealt with a lot of my paperwork, and then my first 18 months of the IB2 next teaching team, especially Lori, who was so fantastic. I also want to thank Mary Markland and Cinnamon Moffat at Hatfield, who really made everything happen for me, especially Cinnamon, who, when I could, whenever I found her, it's like, oh, made my day so much better. And I want to thank especially the Pisco Lab family. We got through so many things in the field, so many great times, good times and bad. Dark mornings and late nights. Couldn't have done it without you guys. It's fantastic, and I love that we still talk work. And now I'm gonna cry, but thank you to Silke for absolutely everything, from that awkward first hug. I'm a hugger, she's not. Found out a little too late. To our plant office, to her making me go diving in New Zealand, Fiji, Alaska, all the eels. I got everything. Our board meetings. Yeah, thank you for carrying on my ship. I wouldn't have been able to do it without you. Thank goodness it went with. Thank goodness we can stand each other at the end of this time. And then I want to thank my coworkers. So I work up in Alaska at a research station, and I would not have been able to finish this PhD without the support and laughs and adventures, especially with Brad and Charlie up here and all the really awesome people I've met. It was a pretty special place, and I can't believe I found myself here. Thank you to all my friends near and far. Some of you have been with me for decades, some of you are new, and oh my gosh, it's been so awesome to get excited about new projects with you guys and then sit and talk about old projects and what to do next. And then I want to thank my family. They probably never thought I'd be in grad school for this long, but they've supported me every moment of it. And here we are, finishing it up finally. So thank you all. And with that, here's where I am now, and I will take questions.
Looks like a great place to work. Let's see, timer. We have time for some questions. So either via Zoom or here in the room.
little bit to add.
Any hands been raised yet?
Nothing
Heather,
obvious.
when you started this project, how worried about climate change for corals were you, and how worried are you now?
I think I was more worried then because I didn't understand, because I didn't know how many ways they had to offset pH stress in terms of carbon concentrating mechanisms. And I mean, they are photosynthetic, and the sheer diversity of modes of survival kind of blew my mind. But I also, I think I shifted from thinking OA was going to be a problem to thinking more about broader picture, maybe temperature, and then just the complexity of the interactions. So I don't want to say this now, but like I feel like I almost understand less because, or I'm worried less because it's so much more complicated that I don't know how to worry about one single thing.
Saw a hand raised.
So we'll come first. If you can hear us.
Can you hear me?
Yeah.
Hey, thanks, Heather. That was really interesting. So not knowing anything about the algae, I was curious if there's a seasonality to their reproduction and settlement, and how that, and you might have mentioned this, how that fit into your sampling design of when you scraped and when you resampled.
Fantastic question. I don't know that we really know anything about seasonality of spore release. I scraped these plots in spring, so they'd have a nice amount of summer and fall to grow in. But I don't, and I do want to look at that seasonality. I do have the data from, at least in Oregon, I have monthly data from the clearings that I haven't been able to look at. And then in Oregon, I have quarterly. So I would really like to look at that seasonal change, and if I'm getting new settlement in summer and then in the winter, it's just that slow encroachment from the edges. So I will be able to look at some of that, but in terms of the species, I don't think we know anything about seasonal, like their reproductive. I think we can get a better sense of like the success of their reproduction, but I don't know that that would actually reflect their biology. That's something I've looked at a lot in kelps, but knowing how little we know about kelp reproduction and seasonality, I think corallins are pretty much a black box on that front, but it would be really, really interesting to work on. Yeah.
We have a question on Zoom that says, which of the 4 future directions you mentioned will you pursue in another lifetime?
I have to admit, I tried to pursue the kelp and corallins chapter, but I think that was chapter 4, and my committee said to stop at that point, which is fair. I have the opportunity to do some more work of that up here, and I think where I am now, I have an incredible diversity of kelp, and it'd be really interesting to work on that question of kelp and corallin interactions, both intertidally and subtidally up in Alaska.
The next question, it says, for your first chapters where you observe high growth rates, did most growth come from the expansion of one or a few individuals, or from many individuals having settled?
I believe, so the sites that had the highest growth rates, I'm just going to focus on these are the 2 sites on the New Zealand West Coast. Those sites actually had opposite reactions on that front. So one site had very low numbers, like very large patches, and very low numbers of patches. So that was mostly encroachment, but the other site that grew the fastest was actually a ton of very small patches, so settlement. Which is interesting because the corallin recruits tend to grow faster than established corallins in terms of surface area. Established corallins tend to grow in terms of like thickening the thallus and strengthening, but they're not necessarily looking for lateral growth, which is what I was measuring. I wasn't looking at crust thickness. So I might not have been able to, so the corallins may have been growing, but the mature ones may have been growing upwards. So that disentangling that is not, like I can't really look at thickness of the crusts without destroying what I'm doing. but the fastest growth rates I saw were through both modes. And that's something I would love to have the information to look at species wise. I don't, because I didn't take voucher specimens. I never actually thought I'd be able to do genetics. So thank you to the funders who made that possible. But I don't know if that was a species specific response or if that was a site specific response. They have quite different geology in terms of microhabitat structure and what substrate the corallines can grow on, and they may have an easier time expanding on one site was kind of mudstone and one was what was it? Decomposed granite. So it was a lot chunkier. And that may have changed how the growth, how they were able to grow. But that's a very good question and a very complicated answer that I don't really have the full answer for.
Next, we have Jimmy Cornelius with a question.
Hi, Heather. Thanks for that really cool presentation. I, of course, have ecophysiology type questions, but I was curious if you think there's any evidence for kind of local adaptation or plastic epigenetic kind of responses to pH. And
Yeah, absolutely.
if some of these places in New Zealand are being exposed to kind of more variable or difficult conditions, if you think that they just may have a more robust phenotype as a result of some of those kinds of processes.
So for the first question, absolutely, the epigenetics are really important. There's a study looking at OA effects on multiple generations of corallines, and they found really rapid adaptation to low pH through, I think it was like less than 5 generations, which is crazy for, like, I don't know what the time was on that, but in the lab, it's a lot faster than in the field. But yeah, there's definitely local adaptation and very fast adaptation in some species. And so yeah, that it's very possible in New Zealand. I was really only looking at species ID. I wasn't able to look at molecular markers that might indicate like different haplotypes or different adaptation or, you know, different functions that way. But I should also mention that the range of species I saw in terms of diversity, I found different orders of species. I'm not just thinking about like different genera, it's like incredibly diverse physiology, even within this super, I think the monophyletic is a super family, which is crazy. And some of these species, and a lot of these species are undescribed. So we really don't even have a good description of them yet, let alone anything about their physiology. And coralline physiology is very young because so much of it requires an electron microscope and decalcification, which can destroy some of the structures you're looking at. Did that answer both parts of your question?
Yeah, no, that's interesting to know. That is really fast. And
yeah,
it seems like good news for them, hopefully.
Yeah, and I think maybe that's one reason I'm less, as Sarah asked, I think that was Graham earlier, how stressed I am about them is that the adaptation is, I don't know that it's in all species, but still a lot faster than I expected.
Yeah, thank you.
So we have 3 more questions. Let's do those and then we'll finish the public meeting.
All right, so the next question, it says, if clearing created an opening for diversity, was any of the diversity VAT invasive or destructive?
Oh God, that's a great question. I mean, maybe I'm biased, but no, in terms of the coralline diversity. I would like to go in and that question about seasonality, I would like to go in and see, I wish I had seasonal genetics, but I would like to look at what species were in those sites that recovered fastest, and if I can identify a weedy species versus, which for coralline sounds very strange. In terms of invasive species, I think Bruce and I talked about this, like I don't think we know anything if there's a coralline invasive species. Definitely with fleshy algae, we know a few, but I don't think we even know enough about species ID in corallines to call one invasive or not. In terms of succession though, there was one site where one of the sites that didn't have very much recovery really became dominated by fleshy algae, and that's something where especially in coral systems, that's a concern because that then crowds out corals and coral settlement. And in temperate reefs, there has been some switch, but it's more pronounced in warmer waters. So I wouldn't say invasive, but I did see and could see in other situations where opening up that disturbance could facilitate a shift to a more fleshy algae or invertebrate dominated system. I saw that at one of my sites in New Zealand where the clearings didn't really recover crustose coral, and they really went to a spirobid worm of all things, took over and unfortunately also ruined some pH sensors. But it was a native species, but just a very persistent and fast growing one.
Following question to the one related to the high growth rates. Were recruitment settlement rates related to the oceanography then?
It's complicated. I think it was more related to smaller scale processes, in that sites close to each other had very different recruitment rates of corallines. But water retention along a coastline does change recruitment, especially of invertebrates, since that kind of plays into the successional dynamics, in that I think it was a previous studies of Bruce that found, plenty of larval invertebrates offshore, but they just were not getting onshore to some of these sites, leaving a ton of bare space. Invertebrate settlement was really low, and coralline propagules could be part of that in terms of water retention against a coast being really important in that settlement and recruitment. But corallines also have a really short dispersal range. It's mostly determined by their height off the substrate and out of that boundary layer. And crustose corallines literally are a boundary layer, so they don't really get very far up. So that makes their dispersal distances, in the order of centimeters to meters, not kilometers, like pelagic or some of the larvae. But water retention against the coast could also impact that as well. But given the diversity we saw, or the wide-ranging species, it's kind of hard to say on that front. But I do think that oceanographic conditions would affect recruitment, but I don't know in what way. And unfortunately, there hasn't been a way to detect larval propagules in the plankton, either with any group that I'm aware of. So that's a bit of a difficult one. But I mean, oceanographic, so big picture, oceanographic conditions definitely affect it, but how and at what scale, I don't know. I think it's extremely complicated.
Right. Last question. Do the thicker, older coralline experience more grazing or more effects from physical disturbance?
I think they may be more resistant to some physical disturbances, in that a rock bashing against them may not remove all the cells, whereas if it's against a really thin crust, you may you may scrape the entire thing off. But in terms of grazing, a thicker crust is also more resistant in that there's more cell layers. So the grazer can take off the top few, or some biofilm and a few layers, but the meristem, where all the new cells grow, is deeper, so there's less chance of damage to it in a thicker crust. Whereas thinner crusts, the grazers are going to get to that meristem region much, much faster. And those new recruits and thinner crusts are also more vulnerable to pH, because they don't have that buffering of the cells around them to buffer the calcification fluid in that area. So the thickness of the crust definitely impacts how they react to a lot of external factors. But again, a lot of that can be species specific due to the structure of the crust, as well as the growth patterns, in that some species seem to prioritize crust thickness and some species seem to prioritize lateral growth. Even though at a young age, obviously they want lateral growth to get as much light exposure as possible, but the physiology of that is quite complicated, but generally thicker crust will be more resistant. And they can get really, really thick, especially in the poles where there's less disturbance. You can get crests that are, 40, 50 centimeters thick. And there's some really cool questions you can ask with that. And the grazers aren't getting that deep, that's for sure. Even in urchin barrens where the urchins are doing their best to get everything out of the rocks.
Okay, thank you, Heather. So we'll end what we'll do with the committee is we'll take like a 10-minute break. So we'll meet on the link that you sent around, Heather, at about 2:20 our time here, which is, I don't know what time it is there, but and I don't know about New Zealand either. It's about 4 hours different. In any event, we'll meet in about 10 minutes via the Google link that you sent. Let's thank Heather.
Sounds good.
And
Thank you.
I'll see you in a few.